Locomotion and movement will be our next lecture, where today we will mainly stress on muscles and their movement, how the movement of muscles actually occurs. Now, before going into any details, we have to know about the type of muscles that are present in human body. These are of three types: smooth muscles, cardiac muscles, and skeletal muscles. Now, smooth muscles are usually found in the internal organs of our body, which are always involuntary, that is, they are beyond our control. We cannot control their movement. Cardiac muscles are found in the heart. They are also involuntary because the heart itself is self-regulatory, as we have already discussed in the previous lectures. Then, third, we have the skeletal muscles, which are always attached to the skeleton or to the different bones in our body. This is always voluntary. Voluntary means we can control the movement of such type of muscles in our body. The muscles attached to your limbs, the muscles in your face, all these comes under skeletal muscles, which we can move at our own will. We can control their movement. So now, today we will try to understand the functioning of these skeletal muscles, how actually skeletal muscles work, how the movement takes place. Now, before going into the details of how the movement takes place, we have to again further understand the anatomy of the skeletal muscles. Now, as you can see here, it is a figure of muscles. Now, this is a muscle. Okay, this is a limb muscle, a hand on your arm, rather. So here you have the tendons, the bones. These are the tendons, and these are the muscles that is attached. This is the point of attachment, connective tissue. We have already learned about it. Now, a small portion of the muscle has been enlarged here. Now, you can see this is a small portion of the muscle. Now, each muscle is made up of bundles, muscular bundles. As you can see here, these are made up of muscular bundles. Here you can see these are made up of muscular bundles. So these are the muscles, and each one made up of some bundles of fascicles, which are called as muscle bundle. So here, this is one bundle. Okay, this is one among the bundle. Now, this is the fascicle. Now, from it, the fascicle, again, each fascicle or each bundle is made up of a number of muscle fibers. These are the muscle fibers. So each muscle fiber, again in turn, is made up of myofibrils. Each myofibril is again divided into segments which are known as sarcomeres. It is units, functional units, how actually the movement of muscle takes place. This is each sarcomere is responsible for the movement of muscle. So this is the brief outline. Now we will go into details of the structure of this muscle bundle as well as the muscle fiber and the sarcomere. So here you get, this is a fascicle or muscle bundle. This is the entire muscle here, and these are the fascicles. These are the fascicles. These are the fascicles. Now each fascicle is in turn made up of muscle fiber. These are the muscle fibers. Now these muscle fibers, which can also be termed as the muscle cell, they have the nucleus, they have the cytoplasm, they have the plasma membrane. Everything is present within this fibers. These muscle fibers, as you can see here, the same figure here, the muscle fiber. They are each connected with blood. That means, with blood means they have the vessels in the form of blood capillaries reaching up to each of the muscle fiber. Now, one muscle fiber. So you can see here, this is one muscle fiber here, which has been shown. Now, the muscle fiber, it has the outer covering, which is termed as the sarcolemma. Sarcolemma, which is comparable with the plasma membrane. This is comparable with the plasma membrane, as we are designating the fiber as one muscle cell. So this you can consider as a cell enclosed by the plasma membrane, which we are terming as sarcoplasm in this case. They have the nucleus, Okay, they have the nucleus. You can see here also nucleus. Here also nucleus. They have the sarcolemma, the outer covering, sarcoplasm, the inner content. Then they have mitochondria. Numerous mitochondria will be present. Then very importantly, they have the sarcoplasmic reticulum, that is the endoplasmic reticulum, which we are referring to as sarcoplasmic reticulum. The numerous sarcoplasmic reticulum will be present inside each of the fibers. They are responsible for release of calcium ions that will actually trigger the working of the filaments. Now again, the fiber. Each fiber is again made up of numerous myofibril or myofilaments. So you can see here, these are the myofibrils. So these are the myofibrils. This is one myofibril, this is another myofibril, this is the third myofibril. Each of them, they are made up of the myofibrils. These fibrils, they contain proteins. And these proteins, they act as units. They act as units to carry out the function of movement of the muscles. Now here this are mentioned I band, A band, Z band. This we will be talking about when we go into the details of the myofilaments, because ultimately we have to talk about the myofilaments. So this is again a muscle fiber. So this is a muscle fiber. Each muscle fiber again has all the cell organelles, sarcolemma, the outer covering, sarcoplasm. Then they have the myofibrils. These numerous filaments. Okay, these are the myofibrils. Each myofibril again made up of the numerous contractile proteins. So here you can see again. Now, this is one myofilament. Numerous myofilaments are present inside a muscle fiber. So this is one myofilament or myofibril. Now this one myofibril, now what is it made up of? It is in turn made up of two types of proteins. Two types of proteins, the filaments. This is actin filament and myosin filament. Okay, so here what will happen? There is one type of filament, that is this blue. The blue that is represented here, it represents the actin filaments. They are present one upon another. The myosin, which are represented in red, they are also present one upon another. Myosin filaments are thicker compared to the actin filaments, and they are always present alternately. They are present alternately, and consequently they give us two types of shades, light and dark. Now you see here, this is a more detailed structure that has been shown here. Now, this one unit we call as sarcomere. Now, what is sarcomere? Sarcomere is one unit of the myofibril. So here, what happens? Now, alternately, a light band and a dark band here. Alternately, a light band and a dark band will be present upon a myofibril or myofilament. The dark band is called as A band and the light band is known as I band. I stands for isotropic, A stands for anisotropic. So alternately, this is the dark band, this is the light band, again it will be followed by the dark band and again the light band. So it will continue. Now, I band, one I band till the next I band. Okay, 50% of one I band, one complete dark band A, and 50% of I band, half of I band. Together, this entire stretch constitutes the sarcomere. This is one functional unit for movement of muscles. Now, the I band, the lighter colored one, it's mainly made up of the actin type of filament, actin filaments, which are comparatively thinner. These are also thin filaments. A band, which is comparatively darker shade, it gives us a dark shade. It is mainly made up of myosin filaments. Okay, it is mainly made up of myosin filaments. Myosin, which are thicker, they are thick filaments, and at a portion, a portion of the myosin, you can see here they are overlapped with actin. Myosin, actin, they are overlapped. So obviously it will give a dark appearance. This central portion is made up of only thick filaments, so it also appears dark. This side also overlaps, so the entire portion appears darker shade. Whereas the portion where there is no overlapping, there is presence of only the actin filaments, will appear lighter. So it gives us alternately light and dark shades. Then 50%, as I mentioned, now the eye band, it will be bisected halfway. Okay, eye band is bisected halfway, and this line which bisects the eye band is known as Z line. It is known as Z line. Then the myosin is also bisected here. Centrally it is bisected, that is known as M line. Then the portion here, H zone. H zone is the portion where there is no overlapping of the myosin filaments. This myosin, they are free. They are not overlapped by actin. These are only myosin. Next, overlapping both alternately actin and myosin. Again, only actin. So here you have to remember three main points. Z line, here one Z line. This side also there will be one Z line. So from one Z line to the next Z line is one sarcomere. So this is about the structure of the sarcomere and the myofilaments. Now let us go into the contraction. Now here again the same thing. Muscle fiber has many myofibrils. So here you can see the many myofibrils here. So this is one sarcomere. This is one sarcomere, this is another sarcomere, this is another sarcomere. So here you have the H zone, then this is the Z line, these are the actins. Okay, so you get it. So here is the Z line. As I told you, this is the Z line. So this is one sarcomere. Then next you have the myofibril has many sarcomeres. So here you have, this is a dark band, okay, dark. Okay, then here is the Z line, this is the light shade. This is a light shade. So here is the Z line. From one Z line to the next Z line is one sarcomere. This is the actual picture how you see it under the microscope. Then this small portion centrally, which is little different in shade. Here is the H zone, only myosin. The next to it is alternately actin and myosin, alternately actin and myosin. Then again light band here. Now, this is the detail again, the structure which I showed you a little while ago. Sarcomere is relaxed here. Now, this is in a, when your body or that particular organ, that particular muscle is relaxed position, then you will find everything is stretched out. Now, when it is contracted, that means when the muscle is walking, then this Z line to Z line, it shortens, and this shortening leads to contraction of the muscles. Now, how it shortens, that we have to understand. And that has been explained with the help of a theory, which is also known as sliding filament theory. Now, before going into the theory, again, we have to know about the actin and myosin. Actin, the thin filaments, which makes up mainly the I band, and myosin, the thick filaments, which mainly makes up the A band. Now, here you will see the actin filament, they are made up of F actins. Here you can see there are two. Okay, two F actins coiled around each other. Each F actin is again in turn made up of G proteins. These are G proteins, globular proteins, and this is F actin. They are wound around each other. Then they have one more protein around it, which is known as tropomyosin. The tropomyosin has certain points upon its surface which acts as attachment points. These points are always masked or covered up by another protein called as troponin. I repeat, actin has two fibrous proteins which are called as F-actin, each made up of globular proteins. Around the F-actin is wound the tropomyosin. Tropomyosin has certain points upon it, certain active sites upon it. These active sites are always masked when the muscle is not working. It is covered up by a protein called as troponin. This troponin can be removed. when stimulated, properly stimulated. This is about the actin, the thin filament which makes up the eye band. Now coming to the thicker filament, that is myosin. Now myosin molecule, it is, you can see here, this is just a small portion of the myosin molecule. So here it is made up of tail and head portion. Tail and head portion. Now this is a flexible portion which can move, okay, which is movable. Now the head portion, it has ATP binding site. That means ATP can act here, can react here, thereby releasing the energy here. And the heads, these are the actin binding site. Now as I mentioned here, here it is specific active site is there. Now whenever the troponin is removed, this head will fit into this portion. That is the main principle behind. The actin binding site of the myosin fits into this tropomyosin active site. That's how muscle will work. So these are the two types of proteins you will find that makes up the myofilaments. Now here you see, which I told little while ago, this is the actin, this blue are the F actins, this orange is the tropomyosin, and this is the troponin. Now whenever this is the myosin with the head and the tail portion, so this way there will be many. Okay, like here again there will be one more, so there will be another one more, so it will go on forming this way. Now what happens, this head portion can bind with upon this active site. Now how it will bind? Here. Now what happens, each muscle is connected, each myofilaments to be more precise, is connected with the neurons. So these are the neurons, okay, they are connected. Now whenever there is a nerve stimulus, whenever a stimulus is transmitted, now this point where the neuron meets with the muscle is known as neuromuscular junction. At this point there will be release of some neurotransmitters like acetylcholine by the nerve endings of the neuron. That release of neurotransmitters will transmit throughout the muscle fiber and stimulate the sarcoplasmic reticulum to release calcium ions. Release of calcium ions. Now what will happen? As soon as the calcium ions are released, they will go and act upon the actin. Okay, they will go upon and act upon the actin. Which part of the actin? Troponin. Little while ago I showed to you troponin. Troponin will get removed. It will open up like opening of the cap of a bottle. So it will open up. As soon as it opens up, what will get exposed? The tropomyosin active site will get exposed. So the moment it gets exposed, and simultaneously this side, the myosin has an ATP binding site. So ATP will walk there and it will stimulate the myosin head to get attached with the exposed part of the actin, tropomyosin. It will get attached. So here you can see this walks, ATP breaks into phosphate and ADP releasing energy, and that energy release will help in binding of the myosin head upon the exposed part of the tropomyosin. It gets attached, so this forms a cross bridge. We call this formation of cross bridge. So the moment it forms a cross bridge, next what will happen? It will move. You can see here, this is bending because this is flexible. The point is flexible, it moves, and so what will happen? The actin will be pulled towards one side. It will be pulled towards one side. So where is this event happening? Here. This event is happening here. So here what happened? So these black ones, these are the myosin. Okay, these are the myosin. So here this is the actin. So what will happen? The myosin will get attached with the actin. And then as it rotates, as you see here, this part, okay, as it rotates, what will happen? They will be pulled. The actin will be pulled inwards. So pulling action will result into shortening of the sarcomere. And that is how muscles walk. That is how muscles contract. Okay. Now here, once more, the active site on actin is exposed as calcium binds troponin. Okay, so here calcium binds troponin and tropomyosin active sites get exposed. So this is myosin head with the Now, myosin head forms a cross bridge with the actin. So here it has formed a cross bridge here. During the power stroke, the myosin head bends and ATP and phosphate are released. So this power stroke, so you can see here it has bent. Where it was here, now it has reached this part. A new molecule of ATP attaches to the myosin head, causing the cross bridge to detach. So here, when ATP attaches, the cross bridge will be broken and it will again go back to the relaxed position. ATP hydrolyzes phosphate, which returns the myosin to the cocked position. That means again it will go to the original position. Further, again when stimulated here, so what will happen? It will again attach, move this side. So it goes on in a continuous manner. As you can see, it is sliding against each other. These two fibers slide against each other. Hence, this is also known as sliding filament theory. So here, this is the relaxed state where you see the H zone. This is the I band. Okay, this is the A band. This portion is the A band. The Z line. These are the Z line. Now, whenever stimulated, what will happen? These are the myosin heads. These are the myosin heads. These myosin heads will get attached to the actin and it will pull inwards. It will pull inwards. So it will pull inwards, which was originally here this way. So now it will result in the shortening of the sarcomere. So that is the contracted state. Now again, as soon as the myosin and attachment of the cross bridge between the myosin and actin is released, it goes back to the original position. So this is how the muscle contraction works. This is what is about sliding filament theory. So this is the formation of cross bridge, the actin myosin structures, and sliding filament theory. Okay, that is all for today. Thank you very much.